Hello, um, my name is Hannah Woolf, and I'm a senior at this high school. And I had an opportunity this summer to do something that I never really expected myself to do, never saw myself doing, and ended up wanting to do again and again and again. In fact, I was just reminiscing through the notebook that I brought with me to work each day. And I was looking at all the notes I took, all the models I drew, and all the designs, and I was thinking, I miss this. I miss the feeling of creating something and discovering something and solving a puzzle which felt so closely connected to me. So to explain what I did is I went to UMass Medical School molecular biology labs, and I worked with Dr. Craig Mello. He was our principal investigator, and he studies a worm. So it's that worm over there. It's not a garden worm, it's a nematode. So it's microscopic. You can see it with your eyes, but you can only really see it through a microscope. And so here's some pictures of me at the lab. This is me examining a gel through UV light, me with Dr. Mello in the bottom right corner. And that's Ahmed Elowa, the grad student who helped me. Well, he worked on his PhD, and he taught me how to be a scientist. Not like science class or science homework, but to be a scientist, to be someone who does stuff over and over and over again, and it never seems to work until it does, and then you know something, and then you have another question, and then you do it all over again. So it's a bit of an exhausting process, but it was really cool. And actually I was inspired to do this talk by Max and Connor, who were in the Excel biology class I TA'd last year. And they came up to me and asked if they thought that what I was doing would be interesting to talk about at this event. And I said, of course it would be. So I didn't really know. I hadn't done it yet. But I agreed to come and do this. And I'm really glad I did, because now you all have to listen to me talk about it. So over the course of the summer, we were studying the function of a gene called F32D1.6, which is not a very nice name, but the reason it had such a long, numerical, boring name is because we didn't know what it did. And that's what we were looking for, because this gene was a gene in that worm that I showed you, and it had no homologs, meaning it had no similar genes in other species. And so it was a mystery. We had to solve it, and that's what Ahmed's PhD dissertation is about. And that's what my summer was about. So here's a diagram of C. elegans, which is the type of worm we're studying. Its scientific full name is Caenorhabditis elegans, and it is a species of nematode. It has a genome 100 million base pairs long and 2,470 protein coding genes. To give you an idea, humans have a genome of 3 billion bases, but we still have only around 20,000 protein coding genes. So in ways, we and this worm are very similar. In other ways, we're very different. But it has the basic parts that a human has. It has a digestive system, a reproductive system, a mouth, and, well, that's basically it. And the 3 experiments that we decided to do over the summer was something called a yeast tube hybrid screening, RNAi by injection and by feeding, GFP tags, and quantitative real-time PCR. And I'll explain what those are. Before I get into that, I'm just going to give you a brief summary of some science that if you were in my TA class, you should know, and maybe I'll quiz you on it later. Basically, the way we and all other living beings, whether it be a plant, a worm, whatever, work is that we have DNA. And that DNA is just a series of letters that comes in a double-stranded helix. And through various proteins in our cells, that is translated into something else that has a language of 20 letters called an amino acid. And when you string all those amino acids together, you get a protein. And proteins are a folded, long, intricate molecule that each are very different from each other and are given a function by the different ways they are shaped. So for example, just off the top of my head, hemoglobin, a protein that you have in your blood, carries oxygen from cell to cell. And the reason it works is because it has little pockets in it that you can actually see if you look at a human-made diagram of the protein to fit that oxygen into. So, and those are called heme groups. So the way a protein is shaped tells you a lot about what it's going to do. And a gene is the instruction code for making that protein. And the transitional quality, the transitional piece to this entire process is something called mRNA. It's that little single-stranded string right over there. mRNA is similar to DNA, but it's only one strand. And it takes something from a DNA molecule to a protein molecule. And mRNA is only one type of RNA that we have in our cells. And it's something I'll talk about later. Okay. So the Mello lab does work on something that's called cell fate. It might even be something that you've wondered about, because you're told that you start as a single cell. But that doesn't make sense, because you're made out of a lot of cells. And so, how does that happen? How does a regular cell become a blood cell, and then a muscle cell, and then a brain cell? And even in a simple creature, like C. elegans, that is a really complicated process that we don't understand. And just a quick video of what it looks like. This is a single-celled embryo of C. elegans turning into a fully formed worm. Whoopsies. How do I play this? There we go. Okay. So you see the embryo, oopsies, sorry guys. So you see the embryo splitting into lots of different pieces. And you can even see the nuclei of each cell. And it eventually divides into one cell and cells. And even at this point, you can start to see certain things. For instance, the bumpy cells are what's going to be called intestine granules, and that's going to form the worm's digestive system. Okay, now you see the actual form of the worm taking shape. It's folding over itself, and it's becoming not just a blob, but a full worm. All the little bumpy parts, the intestine, are clumping together because they're going to form a single part of the worm that you can see right over there towards the right end of the screen. That's the intestine. And you can even see, I'm not sure, a small tube that goes down the center of the worm that's basically its mouth that goes into its digestive tract. So the Mello lab is trying to understand that process. It's trying to understand what makes one cell different from another, and what makes a cell decide to become a muscle cell and congregate at one part of the body, or to become a brain cell and congregate in the head. And even what makes some cells decide to go through a process called apoptosis and kill themselves, because they know that in order for the animal to survive, that's what they have to do. But how does the cell know this? A cell isn't a living being. It doesn't have any way of learning these things. And it turns out the reason that this works is because of all the genes and proteins and letters inside you that are an instruction that's highly complex. To give you kind of a metaphor that you can kind of play with, it's kind of like asking someone to prom. And by that I mean that it's not a simple question of going up to the person that you've liked for 4 years and saying, will you go to prom with me? It's an intricate process of telling your friend Angela that you have to tell your friend's friend Ben that Mike is super cute and you want to go to prom with him. And then Mike has to tell you that he's been actually texting this girl called Sally recently, and because of that, he's not going to prom with you, but he's going to prom with Susie Sue instead, and now he's a somatic cell. So there's a mixed metaphor for you. This is a really cool picture I found. What me and Ahmed specifically were doing, we were studying the gene I told you, F32D1.6, that has a lot to do with what cells become the intestinal cells of a creature. And here you see a single embryo going through the process of dividing, again, But here the intestinal cells are highlighted using GFP, a protein that glows. So it's a pretty cool picture. And this is another picture of the same thing, except it's showing it in a different way. It's showing it looking at the worm through a specific light on a special kind of telescope. And what's really cool about this picture, and I just wanted to share it with you guys, was that when you look at the intestine, which isn't considered a very glamorous part of any organism's body, it sparkles. It sparkles like a star under that microscope. And that's how you count whether the embryo has intestines or not. And it's just like kind of an amazing thing. And that's just one of the things that I learned this summer, that worm intestines sparkle. And here's a cell differentiation map. This is a way that scientists are trying to understand the process of cell differentiation, or cell fate, as I called it earlier. They're trying to figure out the specific path that the zygote takes into the fully formed worm. And this is a really simplified version of it. In fact, if you look at the complete maps of a C. elegans cell differentiation process, they're incredibly long and complicated. And I was going to put one on here, but I didn't because it was confusing, even to me. And so you just see the zygote turns into 2 cells. And from those 2 cells, one forms the germ line, which is the reproductive line of the worm, and one forms the somatic cells, which are everything else, the muscle, the brain, whatever. And so that divides more and more and more, and somehow you get a worm. Okay, so now we've come to the first experiment that I did over the summer, which was the one I started off with. And to me, it's kind of the most interesting one. Oopsies. It's called yeast 2 hybridization screening. And it's a process where you're looking for the function of that gene. I told you F32D1.6. But where do you start? Well, it turns out F32D1.6 has a lot of similar characteristics to other genes in the cell. And in order to find those genes, you use the screening. What you do is you take a transcription factor, which is something that tells a protein that it needs to be transcribed, of a yeast cell. Okay, I'm going to read you something I wrote for this because it's really confusing. Okay. It was invented in 1989 as a way to evaluate protein-to-protein interaction. So it's trying to say, okay, if we have this protein, oh my God, what's going on? Called F32D1.6, and we don't know anything about it, if we find other proteins that react with it, then don't we know something? And so that's basically its job. And it involves taking a transcription factor for a yeast gene and breaking it apart into 2 parts. The DNA binding zone, which is attached to the DNA of the cell that's going to be of the gene that's going to be transcribed, and an active domain. The active domain normally would be part of the DNA binding zone, as you see in slide B. But what you do is you break it apart. And then you take 2 proteins, in this case 2 worm proteins, and you attach protein X, which is called the bait, to the DNA binding protein on the cell. And protein Y, which is actually hundreds and hundreds and thousands of proteins, different proteins that you switch in to the active domain. And that is called the prey. And what we have at the lab are something called prey libraries, where we have basically every protein in C. elegans. And what we do is the gene will only be transcribed if the DNA binding zone and the active domain reunite, as you see in slide B. And that will only happen if protein X and protein Y have some sort of interaction between them. And if the gene is transcribed, and if it's turned into a protein, that protein allows the yeast to live. So at the end of this process, we have a lot of living yeast cells and a lot of dead yeast cells. Each living yeast cell has a different protein Y, a different protein that we were testing to see if it reacted with F32D1.6. And the living yeast cells tell us that there was an interaction between those proteins. And from there, we gathered a list of proteins that were going to help us understand the function of F32D1.6. Okay. Another experiment that we did was quantitative PCR. If you were in Excel biology with me, you learned what PCR is. It's this incredibly useful process that you use from taking one little part of DNA and turning it into millions and millions of copies of that. Quantitative PCR, also called quantitative real-time PCR, is a little more elaborate than that. It's a way of measuring how quickly a protein will replicate itself, or a gene will replicate itself, when combined with other genes. So for instance, F32D1.6, we knew that it didn't work well when combined with a gene called POS1. But what about POS1's relatives? Because POS1 is a protein known as the zinc finger, which has a very distinct structure. So if other zinc finger proteins were combined with F32D1.6, would it still work? And would the PCR still be able to replicate F32D1.6, or would the POS1 inhibit that replication? And the result you get from a computer that tells you what happened tells you how many cycles of PCR it went through. And it shows you a normal curve, which means that if you put such and such amount of protein and such and such amount of all the different ingredients, how much of the gene would you be expected to get? And that's on the y-axis. And then it shows you how much you actually got. How much did the inhibiting gene that you mixed in affect the transcription process of F32D1.6? And we use this to measure the interactions between F32D1.6 and other proteins. Another experiment that we were still working on when I was leaving, because it's very complicated, is called a GFP tag. And it basically relies on this one protein that you find in jellyfish that makes things glow in the dark. It's very cool. And if you can attach that part of the gene that makes something glow to another gene, a completely unrelated gene in a completely unrelated species, then you can watch how parts of the worm where that gene is active and that protein therefore is active will light up. And that tells us a lot about how F32D1.6 functions. Because if F32D1.6 functions, for example, in the building of a gut in an embryo, then the protein will appear mostly in the intestine granules that I showed you earlier, the little bumpy parts. But if it has to do with forming the reproductive system, that's where it'll appear. So through the very complicated process of getting the GFP tag, as we call it, to attach to the strand of C. elegans protein, you find out where that protein is active. Okay. Actually going to go back a bit. The final experiment that we did that I don't have a slide to show you for is RNAi. RNAi is a process that was invented a while ago, and it involves that mRNA I showed you in the beginning. Because if that mRNA is so key to the process of a gene turning into a protein, then shouldn't it be easy to stop that? Because an mRNA will bind to a complementary piece of mRNA. So if you know DNA, every A DNA, adenine in DNA, binds to a glutamine in a complementary strand of DNA. So if we make a complementary strand to that mRNA, it won't be able to be put into the ribosome, and it won't be able to be translated correctly. And so RNAi is a process by which you design your own mRNA complementary strand, and you inject it into the cell. And then you see if you're able to turn that protein off. And the interesting thing about this process is that it involves you doing a lot of work. Because once you send for the lab and you get that mRNA strand, you have to sit up at this giant microscope, pump the RNA into a tiny, tiny, tiny needle that will break if you move it the wrong way. Attach the needle to the microscope, and then slowly, ever so carefully, give the little worms injections. And if you do it wrong, they explode. and their guts fall out, and it looks really gross. And then you've wasted like half your day. So that is an experiment we did that was actually very successful in showing us a key fact about F32D1.6, a gene that we knew nothing about in the beginning. We learned that F32D1.6 is a suppressor of POS1, the other gene I told you it interacted with. POS1 is a mRNA regulator, so it's really, really key in cell fate. In fact, if a C. elegans worm doesn't have this gene, it will die, definitely, because it won't produce all the parts it needs to live. But if you take a negative F32D, a negative POS1 worm, a worm where the POS1 has been inactivated, and then you inactivate the F32D1.6 as well, it suppresses the phenotype, meaning a POS1 negative worm will not have an intestine. But if you then also silence the F32D1.6, the embryo will still die, but the intestine will come back. And so by doing the RNA process on POS1 negative worms, we discovered that. And we also discovered that a lot of relatives to F32D1.6, and by that I mean genes with similar structure, didn't suppress POS1, which at first was really disappointing because that's what we were so sure was going to happen. And we were so sure this was going to lead us to a major breakthrough on a whole new family of proteins. And it didn't work. And so that reminded me of this quote that you've probably heard that allegedly someone asked Thomas Edison about his 1,000 failed attempts to make a light bulb. And he responded, "I didn't fail 1,000 times. I learned 1,000 ways not to make a light bulb." And so even from experiments that you are sure are going to work, and you did everything right, and you controlled every variable and it ends up failing, you still learn something. And in this case, we learned F32D1.6 is different than those other genes, that it must have some secondary function that makes it different than those other genes. And so from there, we had a lot of information to work with and to continue studying. So I've talked a lot about really detailed experiments that involved a lot of expensive lab equipment and a lot of man hours, studying a single gene and a single species of nematode that doesn't really seem to have anything to do with progressing human medical research. And that's the interesting thing about my summer, is that I left really excited by the possibility of science, but not sure that what I had done had actually made a concrete difference. I mean, you go into science wanting to help people, right? You think science is the cure to cancer, it's the cure to paraplegics, or to loss of body function. It is what's going to move us into the future. But I worked so hard and I felt like I hadn't done anything, really, anything concrete. But you have to think about those 20,000 genes that C. elegans has. 20,000 genes is a lot, but it's the same amount a human has. So within those genes are the genes that we need to survive. The genes that are so important in humans also exist in C. elegans. And the same process of cell differentiation of one zygote dividing into a germ line and a somatic line and dividing into intestines and muscles and brain, that same process happens in humans. And by studying each gene that's involved in that process and by learning how that works, we don't just change the way we see worms, we change the way we see everything. And that's what I learned. I learned that you can't always look at the problem just as the problem. You have to look at it as the solution to a much larger issue, a much larger belief that what I did in that lab that summer might actually, in some small way, contribute to something that helps some real people. So I worked in a lab that was across the street from UMass Memorial Hospital. And every day, or almost every day, I would go into the hospital and I would eat my lunch alongside doctors and patients and other researchers. And I just felt like we were all connected by a single drive, that what I did looking into that microscope was in some way relevant to the patients in the hospital and to the doctors in the hospital and to the other researchers in the building where I worked. And so what I learned from the summer really was that a worm's tail is everyone's tail. That what happens to the C. elegans when you eliminate the function of one gene is what happens to a human when you eliminate the function of one gene. And that the progress we make studying the smallest, most seemingly meaningless creatures is going to be the progress that we make in human research and human clinical trials. So thank you all for coming. And that's just a picture of me and Dr. Craig Mello, who was awarded the Nobel Prize for his work in small RNAi research. That was my last day at work. And thank you for coming to watch.